worm tubes that some people are familiar with that live near hot hydrothermal vents on mid-ocean ridges. The ones here are usually only maybe 5 millimeters in diameter and kind of snake across the seafloor. Very small.
Deb Blixen is joining us from the Exploration Command Center at Harbor Branch Oceanographic Institute, Florida Atlantic University, my home institution. Deb's also the Associate Director of our Cooperative Institute for Ocean Exploration Research and Technology, and she was the geology science lead on Leg 1 of this mission.
Right.
She says that pile that we saw before was a pile of talus, probably exhumed rock.
Exhumed meaning brought up and exposed, much like what
let's not even.
what I was thinking. Might be the kind of thing that we would expect to see.
Is my right wing inboard a little bit
Near previous eruptive center, if it's a small one. It just brings up rocks and mud and kind of deposits it on the seafloor. And it looks like the surface that we've been traversing now has been covered by subsequent sedimentation, and the light color suggests that much of it is from these little one-celled animals, foraminifera. We did a gravity core, which is a big pipe with a 1-ton weight on the top of it, attached by a cable to a surface ship that's lowered over the side. You get it about 10 meters above the seafloor, and then you let it drop, and it penetrates the sediment. We did that on the top of one of these seamounts, and brought back the top meter of material was completely foraminiferal shells.
The pressure is balanced.
Virtually nothing else except a little bit of volcanic ash. So the accumulations on these, especially at depths like this of about 3,300 meters, can be extensively dominated by these foraminiferal shells, and they are tiny. They are maybe millimeter, couple millimeters at most in diameter.
Do you want to do a quick zoom on this rubble? I'll just kind of pan
Let's see if there are any critters attached to the rubble. Almost every time we zoom in, we'll see something living on the seafloor. It's just amazing to me.
There's a white upper left
and fascinating.
Bigger rock. Now it's straight left lasers.
White,
yep. Tip on us.
We were just chatting online in our chat room about what's really one of the many things that's really cool about these dives is that they're basically rolling invertebrate zoology courses.
I'm learning a lot.
either giving some of us who learned this, I won't even tell you how many decades ago, a kind of a refresher on what it was that we learned. But for many of us and many of the viewers out there, some new information. This is really incredible because for those of us who teach some of these courses, what's that little? Looking at something
on the right?
Above the
rock?
Laser dots. Right in the middle there. What is that?
Oh, the little white thing on the rock.
Yeah, well, in that little crevice in the rock.
Yep,
Yes. What is that?
It has radiating something.
It certainly does.
Sitting down.
Oh.
What do we think that might be?
Sponge.
Could be.
We saw some stalked sponges on one of our dives that had a hair-like radiating.
A couple days ago.
That's what it is. It's the same sort of thing.
It's a carnivorous sponge.
I love that. Carnivorous sponges.
Yeah, these are sponges that are adapted for life in the deep sea. They don't filter out food particles
from the water, right?
Oh wait, now let me look at this closer. You know what? I'm not sure.
Okay.
I have to tell you that looks more urchin-like.
Yeah, Tara says it's an urchin.
Yeah, it's an urchin.
Chris says it's an urchin.
Oh wow.
And Robert Carney says it's a sponge.
Teeniest. Well, now looking at
the spines on this, if it's an urchin.
Yeah,
Holy smoke! It's like 15 times the diameter of the center of the critter.
Wow.
Well, maybe
Wowzer.
10. But
Wowzer. No,
nevertheless,
no, they're really
they are
long.
long and just hair-like.
Wow. Wow.
Okay.
So is this delicious if it's a sponge or this?
No, it's not a sponge.
It's
It's not a sponge. It's not a sponge.
Okay.
Urchins are on.
So we all think it's
Now,
an urchin now.
Les is also thinking of
Siphonophore.
benthic siphonophore. I don't think so. I think it's an urchin. I think it's an urchin.
Wow,
Right
it's so
edge of the
tiny.
screen.
It's really
You
hard
see
to
it?
determine.
Center right-hand edge.
We do have an echinoderm
yeah.
specialist actually in our participating in this,
yeah, let
Allison
me
Miller
just
from the
do.
University of Guam. So we're
How much more does
asking
Sirius
her
have to
for
move
her
there?
guess.
It's still gonna be moving
I don't even-
Oh wait,
for
no,
about
she's
4
actually
minutes.
away for a while.
Oh, too
She's
bad.
doing her day job.
Do a quick,
Ah,
do a quick look at this, yeah.
well, we are too. Okay.
Nolan Barrett, a summer intern at Harbor Branch.
So we're
is telling us
seeing
it's
something else
health
here, but
curious.
it's going to be a quick snap or something
get
down
close to
here.
the pilot.
Our videographer, Roland, has a very nice screen, and he's able to see things before most of us
Sirius
can.
still has about 20
But
meters.
he
Yeah,
has
straight
seen
left
something
lasers
down
to the
here.
left corner.
Gonna let Shirley write down what people are saying about
Keep it in the bottom left
that
corner.
interesting critter.
Oh, I went past it a little. Oh, I see it there,
The pilot sees it. It's under the rock. No, it's not.
Center
yeah.
screen.
Yeah,
It
there
is under the rock, and it looks like...
Another little urchin.
Oh, there we go. Another urchin. Let's see if the spines are as
Wait,
long.
Let's look at that. Can we see? Yeah. Oh wow.
That's a
He's a
pretty
different
one.
guy.
That has a bigger central body and shorter
no.
spine.
You can see the. We can
You
see,
can see the top
disc,
of
yeah.
it, the disc, and the. What is the opening on the top, or is that an opening?
Yeah, it is.
It is. That's
Yep.
its mouth.
Well, I'm thinking it's the other end.
Okay, but
So
look,
it has an oral
Yeah.
surface
The
and
white-
an aboral surface.
Yeah, let's
the
go
white-
And we're also
blebs
seeing.
at
Okay,
the
we're also
base
seeing the
of-
blebs,
and
which
those are the gametes.
gonadal tissue.
Excuse me.
Gonadal
Those are
tissue.
Gonadal
Right,
tissue.
that's what Chris Ma
010.
has been telling us. Is and
that's
actually that's a
interesting.
good sign because it
Yeah, it means
indicates
it's
that the
healthy.
animals are healthy enough that they're
Yeah.
able to produce
To grow.
Yeah, procreate.
reproductive tissue.
20 meters ahead.
Well, we are a lot shallower than we were yesterday. And even yesterday we saw, I think, 2 animals that had those same white blobs down close to their discs. Okay.
We're calling this a glass urchin.
a
Isn't that
glass
beautiful?
urchin, yes. And it certainly looked like it. It was really nice. Well, we do have a lot of sediment on the top of this mud volcano, which suggests to me
Yeah.
that at least this portion of it has not been covered by mud flows of serpentinite, and I will explain what that is in a bit. But that is
What
makes
the?
up.
Look, it looks like a treasure chest.
I don't think it's
definitely
anthropomorphic, but it is a nicely
And wait, what do we have on that?
Nicely
Looks like it might be-
fractured
is that
rock
s-
with a
is that
crinoid.
Mhm.
Oh, but the surface of it is fascinating. It looks like it has a stair-stepped fracture in it. So we are looking at the front face as the tall black area, and then it has a little platform, and then it has either, I can't really tell, it either has a ridge on it that the crinoid is on, or that's another stair-step. That's a really strange-looking rock.
Do you want to collect that?
I would love
Roland.
to collect that rock. This is our videographer making jokes. He's been teasing me. I would love to get that rock. I'll come back later, possibly,
With a bigger ROV.
with a submersible and
And-
anchor it to the seafloor. I don't think anybody could pick that rock up, except maybe a dredge. Nice urchin.
So we are
Crinoid.
Crinoid.
degrees
Urchin
off
crinoid.
Of, ship's heading.
Same phylum.
I wasn't saying it was an
phylum.
urchin. I was
Same phylum.
burping.
Same phylum.
Yep.
Beautiful. Very pretty.
That has very, very
That's
tiny
different,
cirri.
different cirri, yeah.
They're
Oh,
real
and you
short.
can see those little
Now,
pinnules.
what is
those
the blob
pinnules.
right in the center that's sticking out from the top of the disc?
Okay, so
That's a mouth.
Yeah, my guess is
Okay,
that's,
finally got the mouth.
I think so.
And it also has bulbous
We should be
ends
close to
to
done moving with Sirius.
the arms, on its arms, close to the center of the body. So these guys are doing pretty well. Gametal tissue? Gametes? Gonadal tissue.
No, gonadal, but I'm
in the center,
I,
the
you
bulbs?
know what? I can't say. I can't say.
Well, I
Not gonna
certainly
get-
can't. Just guessing. It's fun to guess when you know nothing about stuff. Sometimes you get it right. So what are we seeing? The ray structure is an anal cone, says Scott France, not the mouth.
Whenever you're clear, video.
Okay, as I pull out on the top right corner, I'm going to momentarily pause.
So the other
until
day,
white object maybe
Chris Ma was
comes up.
talking about, was telling us about
See
an
that, yeah.
anal sac, and I guess is that kind of, and that, you know, not all of the echinoderms have those anal sacs, so this might be similar. Okay.
Oh, this is great. Scott
Okay.
France says, oh, let me read this.
Okay.
Since the mouth is down in the center of the disc as well, the anus is raised to be sure that the wastes are not introduced back into the
next one will
mouth,
end up being
which is a
north,
very
so if
good
you guys
idea.
wanna make your way over
We're
there.
having an online chat about
He seems
the
pretty
structures
steady.
about this
So I think
topic
he's passing me
of
by.
this topic
Oh, yeah,
of
he
waste
stayed there.
disposal.
He shouldn't be moving.
And I also got a really good view at the top of this rock, and it has a triangular
Where did it go now?
prism sitting on the
Oh,
top of that
yeah,
big
okay.
rock
I thought so, but I
that the
lost sight of it.
crinoid was on. And that's fascinating. I have no idea how that sort of thing can happen unless that rock got dumped on top of the big flat rock.
Yeah,
But
I'll
I
just
don't
drag
think
right
so.
ahead into center of Sirius once I get this. Okay, you want to do a zoom here?
So I will leave the explanation to
This is
Deb,
stop.
our leg one geology. Oh, there's a sponge. There's a
It is.
sponge.
It is. So we have
The
a
geology
beautiful.
lead on leg one. Now, what's the second?
Right, we've got
creature?
we've got a hexactin. It's like a
Oh, I can't wait till we
hexactinella
get the zoom.
sponge.
So it's got something really wonderful.
Well, it's got.
What
It's
is
got
that?
a
It's a
couple
different
of, no,
type
it
of
looks like a couple.
sponge.
Well, no, that's a sponge. It's a
Yeah.
hexactinellid sponge, but it's
That's
got.
not part of the
No,
one with the bulb on top. But
No.
where's
The
its-
thing that's attached to it is not a sponge. It's a crustacean of some sort, and it looks. And I'm not sure if there are actually
Okay.
two other things there. Let me see.
That's what I'm
A
wondering.
pregnant mysid
Holy-
is what Les Watling
Okay,
is telling
what
us.
is a mysid,
It's another
Les?
crustacean. Yeah. But you could, but that, and also, Chris, that hexactinellid looks a little bit different from what we're
I love this.
what we found.
Pregnant
Certainly different from what we saw yesterday.
Not sure.
Yeah, now that
Tilt
I'm
up
looking
over
at
this
it,
one inside.
I would agree, Shirley. I was just typing in that it was the same thing, but now I disagree. It does
Okay.
look different.
Thank you, Scott. A mysid is a possum shrimp, and it is going to have progeny, except that it's left.
Okay, so
Okay.
Roland's pointing out that, this is kind of interesting because, and Chris, I don't know if you can see this, but the way those the spicules are arranged on the surface, they're kind of in tufts, so that might be diagnostic. So when we show this image to our friend and colleague, an expert in hexactinellid or glass sponges, he might be able to be able to better identify this based on the fact that these spicules are arranged in discrete tufts over the surface of that sphere.
Okay, and back to this very interesting possum shrimp. Les Watling is typing in that the brood pouch under the thorax, just in front of the long, thin abdomen, contains something called yound, Y-O-U-N-D, about which I know absolutely nothing.
No. It's young. He just mistyped it.
Oh, young.
Read down further.
Oh, well,
Read
sorry.
down further.
I thought that was a new word for me to learn. Okay. Sorry, Les. We all do this. We're typing so fast, when we see things and we get excited that any letter that might be close to the one we're trying to hit often shows up instead.
Basically, these isopods and amphipods and mysids, these are all paracrids. Brood their embryos in a chamber below the thorax.
So then, when they're born, do shrimp have the same morphology as the adults and just shed their exoskeletons as they grow?
They go through
larval
different stages.
Okay,
yeah,
so they have a larval stage. All right.
Yep.
Yeah, it's important. You can see it in your.
It's really fun reading the chat.
Oh, I can. Yeah.
Sorry, you folks can't see it all. It's hilarious.
It's probably better that they can't see it all.
Yeah, after
Some of the sidebar conversations we're having are actually make us sometimes belly laugh.
That's
how
true.
funny they are. So thanks. Thanks, colleagues.
And in fact, if all these folks were out here on the ship with us, we'd probably be doing the same thing. And research cruises that we go on, other than here on NOAA's Okeanos Explorer, are kind of like that, with lots of scientists
almost nothing
clustering around one another and just getting all excited together. Sharing experiences of discovery are some of the best parts of the type of work that we do, and so we're delighted to be able to share this with all of you on shore.
Yeah,
and such wonderful
right.
high
And we have,
def.
so it's in addition to just Patty and me, who are just the science leads on board the ship, we have a large crew of scientists, shore-based scientists, who are also part of our science crew, and you're hearing from them, many of them today. So what's really totally cool for me is that I'm basically getting to cruise with a number of scientists that I'd normally not have the opportunity to cruise with, including
Shirley
Patty.
and I've never sailed together before.
But also we're cruising with the shore-based scientists who are very much an integral part of this science team. So it's a great way to explore and to take advantage of the expertise that exists worldwide and is being brought into this mission.
Yeah.
What are we seeing here? Look, some more of those grooves, Patty. The furrows that we saw before. Well, they're those similar furrows that we saw before that you were questioning what they were. So we're seeing, more of those.
Yeah.
Bridge, Nav.
Diving in the Challenger Deep
Hey, we're going to put in a
with the nearest remotely operated vehicle in 2009 at almost 11,000 meters. We saw grooves just like this on the flat sediment plain at the bottom of the Challenger Deep, and that was at 10,924 meters. Whatever is making these kinds of things, I would sure love to know what it is.
We got another sponge there. Looks like another stalked.
Yes, we do.
Yeah, hexactinella. And does it have another? Is this another one?
It has something on it.
It does.
It does indeed.
Looks like it has another one.
Oh, wait for the zoom. Wait for it.
Wait for it. Wait for it.
Wait for it, folks. Oh, now that's like an anemone.
Scott, your
That's an anemone.
dreams have been answered. It's not an octocoral, but
It's an anemone. And when you guys see it, folks on shore have a bit of a delay. Maybe sometimes if people are dialing in by phone, it could be up to 10 or 15 seconds. Usually in the expedition control centers, it's 2 to 5 seconds. Exploration.
Command.
Right. Command centers.
Long.
This is more like my nightmare than my dream, Shirley.
Hey.
I know, Scott. Tell us about this. What do you think?
Beautiful.
Well, Deb did suggest that this was Relicanthus, and that may very well be the case. Relicanthus is the scientific name for a group of what, until recently, were thought of as sea anemones, but molecular genetic work suggests that they're actually very distantly related the sea anemones. So they're in the same group. The Hexacoralia has the sea anemones, the stony corals, and the black corals, but they are grouped unto themselves, and they have these tremendously long tentacles that I believe
Okay.
can extend more than 3,
Yes,
4, or 5
please.
meters. I forget the exact length, but they are long.
Yeah, these are really, really long.
You can
Yeah,
see
go ahead,
them
Chris.
as hair-like.
It's really weird. So on that, in the Northwest Hawaiian Islands, when we collected one of these sponges, this one does look like the weird Calophacus or the Caledoniella. But on that seamount in the Northwest Hawaiian Islands, just before we collected the one sponge we got like this, there was a bunch of them that had Relicanthus holding on to their stems like this. Now, what the heck is going on the other side of the ocean? We're seeing maybe a slightly different version of that anemone, but the same anemone hanging onto the stalk of this one.
Well, Chris, I will tell you
I
that
wonder if part of it has to do with how high up above the bottom the larva wants to settle, because it doesn't seem like there are any walls or really big rocks around here for this anemone or this Relicanthus to live on. And before the observation that you're talking about in this one, I don't think I'd ever seen it on anything other than a fairly steep rock wall.
Okay, I have
Dead
to.
on there, Scott, because
gotta tell them.
all those really long tentacles, if they're all getting bashed against the substrate or something, they're going to get damaged frequently. So because they're so long, they need to get up and off the bottom a ways.
So Andrea Cotrini wanted to know if this anemone was collected on your expedition, Chris.
No, because when we decided to collect the sponge, we started looking for ones that had this Relicanthus on it, and we couldn't find any more. We found just the bare sponges. It was getting toward the end of the dive, and we just grabbed one.
Okay, so do you
The
know?
only problem with this collection is that this would be the third of those type of sponges that we would get. So we really wouldn't be collecting this for the sponge. We'd be collecting it for the anemone.
Should I stop
Yeah, I
Correct.
agree with that last point, Chris. This isn't a sponge collection. This is a Relicanthus collection.
Yeah, this bridge.
And
Hey, let's go ahead and hold
for
here,
that
please.
reason, I think it's worth it. As far as I know, there is only one species
Are you
described
white?
so far, and I believe that is a bluish-purple one, and it's bigger than the one we're looking at here. Maybe someone in chat room, Andrea, can correct me if I'm wrong on that.
Well, we got 20 meters out of that move, so.
Okay, so is the decision to go ahead and collect because we can't
Can't wait.
sit for too long?
No, we're.
It's looking like the consensus is yes.
Okay, I'll move.
I would recommend collecting because
Okay.
this is a perfect opportunity to get one of these anemones in perfect shape, because you're actually going to grab the sponge, not the anemone, and get the two of them into the box. So I can't think of a better opportunity to get a marvelous specimen of that anemone.
Okay, we're going for it.
Pilot, what they're particularly interested in is trying to preserve
Sounds
those
like we're backtracking,
incredibly
but I can
long,
do it.
delicate
So 10
tentacles.
meters,
So I'm not sure,
180
but can
degrees,
you
0.2.
maneuver
Correct. We're gonna
to
take a
part
specimen
and
here.
get the
Okay,
minute
roger that.
underneath, kind of the backside of where those?
Yeah, I think if you get the. I think if you
where the
get,
tentacles
I think if you
are
get it from the
extending.
stalk, it's going to be okay. I think if
But low down.
Yeah, low down on that stalk will
Yeah.
be fine.
Let's do a
Okay.
port inner outer, yeah.
Pilot,
Okay, I will tell
from the
you
video
folks who are
chat
interested
is
in this
if the
specimen, although I
tentacles touch your
never
arm,
knew what it
then
was.
it's probably
I have
attached to your
seen anemones that
You may
look,
wanna,
to
pull
my
that around
eye,
to the
which
right
take that
so
with a
the
grain of
claw
salt,
comes in at a different angle.
very much like this, but a
Leo.
darker red
Take that advice.
on some of the tall carbonate chimneys that form at the active vents on some of these mud volcanoes at about this sort of depth elsewhere in the Mariana system on these volcanoes. And they only have been seen within the 100 kilometers to the west of the trench axis. So from the trench axis back westward 100 kilometers. But I'm curious about what they're doing there, because the fluids coming out of those chimneys have an extremely high pH. And I just wonder how an anemone that is, getting stuff out of the water, and the chimneys are really porous. the fluids are moving through the entire body of the chimney. How could an anemone survive in an environment like that?
Co-pilot will keep an eye on that tether versus the swing
They're
arm.
blowing away.
Thoughts are
I'm seeing
welcome.
more of the back, and if you want, I can zoom in to
No, that's okay.
give you a better visual.
This is some very soft, kind of clay-like sediment here.
You can take the direction that your dust has traveled, and that would be the way the current was going. So that'd be the direction the current tentacles are flowing.
Okay. Yeah, want to get a quick zoom there?
Look
Yeah,
at
they'll be
those
going
tentacles.
generally away now.
Wow.
Okay, you can come wide. Thank you. So you back the ship up a touch now?
I did. It probably hasn't
So
taken effect just yet, but it
now what the pilot's done is, as you can probably see, moved around to the port,
you
to
have
the
not
left
pretty
side, so that the
Okay, yeah, it should-
tentacles
it should
now
be
are
shorter, okay.
essentially flowing more away from the ROV, and he will extend the manipulator arm and try to grasp the stalk of the sponge down below the anemone.
So actually, you just want as much of the stalk as we can get
Yes.
Yes.
to avoid the anemone? All right.
Correct.
Okay, thank you. Arms live. Yeah, that's, I just rotated more than 90 around it and it didn't really change direction, so I think there's such a small bottom current, it doesn't really, can't really choose the current direction.
Yeah, usually as the pilot is doing a delicate maneuver like this, those of us on the ship keep quiet, let him
Okay, I want to
concentrate.
go in a touch on Mini Zeus. That's good for now.
So Scott points out that if the anemone does not contract when you go to collect it, Carl,
Yes.
that the tentacles are going to still be trailing a long way. So, you know, don't worry too much about trying to stuff it all into the bio box. Just drop in the stalk with the anemone and close the box, and we'll just keep our fingers crossed that the anemone will withdraw them. Because we could spend a lot of time trying to stuff the tentacles in the box.
Do these are these tentacles? Do they have stingers? Do you guys have to be worried when you collect them out of the box?
What do you think, Don? Looks pretty centered in the blades.
Yeah, Roland asked if the tentacles have stingers. Like all cnidarians, they will have nematocysts, but like with all of our samples, we handle them
With gloves. Plastic
Actually,
gloves?
I'm double gloved when I first pick these up. I have my plastic gloves, my latex gloves on, and then I have long.
It's like a daisy.
I have gloves. Oh wow, that's pretty. I have gloves on that go
Yeah,
just not up to
Zoom.
my elbows. So
Okay.
Yeah, Zoom may tell us if the tentacles have contracted.
Yeah, take the Zeus. Come on up.
Hard to tell. It looks like maybe they
Do you want
have
to zoom on the
a bit.
tentacles?
Oh, look at that. Looks like
So they have not contracted, right?
I
a
think they have
little
a
bit.
bit.
They have a bit.
Yeah, some of them
I
are
have a
still
bet.
long, but many of
It's
them
got
have
the port
contracted.
box opened up.
Well, this
Could
is going
he
to...
just rotate just slightly so we see the back of that sponge while he's got it in his hand?
Pilot, can you rotate just
Or
slightly
manipulator, I
so
mean?
we can see the back of the sponge?
Yeah,
can
we want to
try
just
as
be
I go
really
away.
careful that we don't lose it. I think we take a chance of knocking it off if we do that, Chris. We do have some good video from yesterday, and the specimen that we have that we collected as a paratype yesterday is
if you
in
want to,
very, very good shape.
Yeah, zoom out on Mini Zeus.
Okay, and I can see what I think I need
Yeah,
to see at this point.
right.
So it does look like a caulophacus to me.
Yeah, it does. It definitely does. And when you see the actual sample, you'll be even more convinced.
Rotate, stick with.
So this is going in
That's the
the
plan.
port outer box. Oh, clever pilot. Ah. Looks like those tentacles are all pointed in the right direction. They are incredibly long though, aren't they? You can see them against the white of the box. So I think, folks, if at home, if you go to feed 3, I think you can see
We'll
the
just have
bio
to close
box,
the box,
and the specimen in the bio box below where the manipulator arm is. And then what will happen is the pilots will slide that entire sampling tray back under the vehicle, and that will keep
Okay.
it closed
so
and
fragile.
keep seawater, ambient seawater, in there.
Yeah, there's a lid that'll go on that box. Oh. Wow. Really?
Okay.
Oh, real. Oh,
Spectacular
my
collection.
gosh. Really, really nice.
Close her up.
Spectacular
Really
collection.
nice, Carl. Thank you. Thank you, pilot. If you haven't noticed, all of our pilots are exceptionally skilled, not only at flying the ROV and handling the manipulator arm, but also doing these collections. Really awesome. Thank you very much, pilot,
You're welcome. That was a
co-pilot,
fun one.
videographer.
So for the preservation of that anemone, I think you had some guidance on that. Is that correct? I think, but I can't remember what it was, Shirley.
I believe that we want to put that in, well, we're going to want to preserve it a few ways. So I think what I will probably do is
Thank you.
put it in formalin. I'm not sure if you want me to inject
I wasn't sure if
the
the ball would start
what's that
to
called?
rise when I dropped it.
gastrodermal cavity with formalin. I'll do that as well if I can. I'm also going to snip off a
Okay,
little
hydraulics
tentacle,
are
put it in
off.
95% ethanol, and
Or
I'll take a
hydraulics are
small
off,
piece
yeah.
and put it in OGL buffer. How's that sound? We have one more tube of OGL buffer. The other alternative would be, instead of putting it in OGL buffer, I can put it at -80. So I'll
Backwards.
wait and hear from our experts on shore about the best way to preserve this to be able to get morphology and molecular biology.
Yeah.
Right where you're headed.
Oh, that's the wrong way, though. Yeah.
Yeah.
Yeah, you wonder about the furrows being in a line and wondering if something came down and just scooted along the seafloor.
So these samples that we're collecting are being deposited in archival institutions, into museums, so they are available for the entire scientific community to study. The biological samples are going to the Smithsonian Institution in Washington, D.C.
Try it.
We're taking subsamples of most of these biological samples and preserving them for molecular work. Those samples are going to the Ocean Genome Legacy at Northeastern University, and as Patty mentioned earlier, the rock samples are going to Oregon State University. And anyone can make a request to borrow and study these samples. There are request forms that are available. You make the request to
the
Okeanos Explorer website.
Ocean Explorer website, to Ocean Exploration and Research.
I've been playing with it. I think it's pretty good there, but just if you let go for a while, like now, you slowly float up. So.
Okay.
But usually in stuff like this, we'd want to fly maybe just 1 meter up so we can really see the little creatures.
Okay.
Are you going for that?
Yeah,
white
we
thing
can
for
see
video?
some more, like the white dot over to the left is
trying to get
another
1 meter right
hexactinellid
go
or
over there.
glass sponge. So these are
Maybe
pretty
it's the
common.
same
We
one.
collected
Oh, is
one.
it?
Yeah, we collected one
Ah.
yesterday.
Should we take a look?
We can.
We're going that way.
We can do a snap zoom. We don't need
If anything,
to
it'd be
stop.
backwards.
Terry?
So that was the last move.
And I'd recommend really soft landings. This stuff is very fluffy.
I will do what I can for that.
Science requested only a flyby on this guy.
Okay. Well, go ahead.
Oh, there we go. Yeah. I'm just wondering if there's anything on it.
It's got something growing on him.
The tuft at the top. I don't see any extra critters.
Go
So
ahead,
this has
Leslie.
got these little tufts, at least one of them on the top again, Shirley, and I was looking at the
Give me a
top of
second.
the one
I
we
will
collected, and
Measure
if
that
it is
and then give you
Caliphacus,
the
it's
time
something
there
that
at
we
the
have
speed.
never seen. And so this, again, yesterday's and today's specimens are, I think, quite valuable.
Okay, video's clear with this.
Okay.
So the total length
up
of
then.
the in terms of the distance we want to traverse on this dive track is about 720 meters, and we had about 4 hours for the dive. We're
down
just
so far
checking
and
with
then
the with nav to see how far along we are on our dive track.
Right.
Those of you watching at home, you can get an absolutely stunning view from the Sirius ROV of the Deep Discoverer on the bottom at 3,308 meters. I believe that's on your screen too, or feed too.
And it is a relatively flat seafloor.
Which we knew when we planned this dive, because we're looking, the depth range was really
I'll wait till they
only,
finish.
33,
continue
30
on here.
meters to 3310. So we expected it to be to be flat.
And what we're searching for here, we are diving on a very unusual
right of lasers?
seafloor feature. These are known
around here?
active
Or a
only
little further?
in the Mariana
Oh, okay,
system,
gotcha.
only in the Mariana
Right, over here?
region, and
Yeah,
only west
Copy that.
of the Mariana Trench
May just be
for a
settling.
distance of up to about 100
Shape.
kilometers.
You wanna do a quick zoom?
So from the trench axis westward 100 kilometers, dotted
might
along
just be
the
sediment.
seafloor are enormous
Yep.
mud volcanoes that are
I'll fly forward just a
erupting
little bit.
mud. So these are not like the volcanoes of the island arc, but rather these are
If anybody
erupting
sees anything, let me know.
cooler material. They're slightly warm, maybe 10 degrees C. We
Yeah, copy.
drilled into one of these enormous mud volcanoes once with the Ocean Drilling Program's JOIDES Resolution drill ship. And down about 300 meters into the seafloor, we got a temperature of about I think it was a maximum of 350 degrees or so. Excuse me, 35 degrees or so. So very warm. I was thinking 3.5 with a little 0. For the degree sign. Pardon me. At any rate, these things are erupting the ground up product
See some rock structures
between
coming up.
faults
Below this
beneath
ground.
the seafloor. So
Are we in a move right now?
when earthquakes happen
I was
and
wondering if you
rocks
could go ahead. Are you ready
grind
for
against
move?
one
Sure,
another, actually right at
Alright,
the fault
ROV target
contact,
bearing is due
the rocks are
north. What do you
ground
think?
up. And
you
fluids
wanna do, we'll
are
do
being
40 meters
squeezed
again?
out of the
Copy that. I
downgoing
have
Pacific
a couple
plate.
rocks like we talked about up ahead, but
And what
Let's-
happens is
let's do
that
30s.
Okay.
these fluids
That'd be a
are
little-
light,
And the next watch.
lighter than the stuff around them, of course, so they rise into the cracks above them,
See the,
and
series
they react
cameras at
with the ground up rock
45,
there
so-
and change the mineralogy,
that.
change the
Well,
mineral
partially has
to
the stream.
I see
incorporate
something.
some of the water.
Yeah,
Therefore,
let's go
those
for this.
minerals expand.
the one that's
They get
floating or the one on the
new
ground?
mineral
The
is,
one on the
is
ground, I think that's-
larger
up about here.
Yeah.
and
Copy
is
that.
less dense than the surrounding material. So as the fluids rise, it kind of mobilizes this
Bridge,
mush
Go ahead and give me a
of
quick zoom
ground
if
up
you'd like,
rock
just so we can
and
see.
fluid, and we
Something there.
we
Should
see
move.
these
Okay, I'll head in there.
30
mushes
bearing
erupt on the seafloor
north,
to
60
form
degrees,
seamounts that
Yes,
can
I
be
do.
as much
Up
as
ahead.
50
Yeah, that probably
kilometers
is
in
part of it.
diameter
Okay.
and 2,000
I'll
meters
go check out
tall.
that then, and I'll watch these rocks.
So the rocks that are brought up by these things can
Little
be
tilt
from
down
the
on the camera.
mantle
Okay.
of the area between the trench axis
It
and the
almost
volcanic arc,
skids
that
and beams, but
200
then the
kilometer
landing gear
wide
also.
Okay.
patch of
I'll get up
seafloor
ahead
we
real quick
call
just so I can
the
get around this
fore
rock.
arc. In other words, it's on the trench side of the active
Oh,
volcanic
thank you.
chain of the
Yeah, I better not
Marianas.
pull too far, huh?
Yeah, it's good to.
And that whole
well, you can
area
look
is
at these and
covered with
the ship
these
hasn't started
seamounts.
moving, you can wander
I'm
anywhere
starting
east
now
or
with better
west.
bathymetry
Okay.
to be able to
So
count
you
how
don't have
many
to
of
really
them
move
are,
north.
and it's like 20 or 30 of
So that's
these
the view
that
for landing
are
generally?
very
Yes.
obvious,
Yeah,
and then
and
there
if you
are
wanna-
very much
if
smaller
there
ones
was
that
some-
just
like,
are
if you wanna
beginning
get a
to
really
form
good
at
view
the
of that
base
rock,
of some
since
of
it's just flat
the
and there's no
fault
current, you can
walls
just
that are
get it
exposed on the
basically
seafloor. So
exactly
there are
between your
lots of these
skids.
everywhere
okay.
out here.
And you can use your mini Zeus to do that if you want.
We're seeing a lot of
Something white
rocks.
diagonally up left.
And so you can set
Mainly,
down, that'll be just super
not
close.
really, I wouldn't
I'll
say
go-
these
I'll
are
have to get that one that's
outcrops,
further
but
away 'cause I
rather
can't- I don't have enough
collections
leash,
of
I don't think.
rocks.
Will that be okay?
Yeah, you
I'll
want
set
to
down right here.
see what it is.
Sure.
What happens when the mud volcanoes erupt is that they, ooh, there's a critter. Let's look at the critter
Nothing, right?
first. Ooh,
Yeah, that's
is
the
that
thing I
crab?
saw for Carl
Gal-
earlier.
Is that a
Okay, I'll go
squat
ahead and
lobster?
set down then because I'm not going to have too much more.
Squat lobster, I'm
It's not going to
guessing.
be a great zoom, but
There's also a starfish buried right between your skids.
right there.
Now came into view, yep.
Do you see star shapes in the seafloor there? Or am I crazy?
No, over to the right of the laser.
Yeah.
Okay.
Yeah, I wonder if those are from worms that, you know, flick out their gelatinous
That guy?
arms and then pull them in and then eat
Oops.
all the things that get stuck to them? Or is it a buried sea star? Mm-hmm.
Do you want to just zoom in just to see if I'm stable? Copy that. Okay. Anything here? Not really.
Oh, wait.
Hmm?
Look up. That's something covered in.
Now we're looking at the pile of pebbles,
Something's in there, huh?
Yeah.
and you zoom toward
Yeah, it looks like it.
Little
toward the
tilt down.
bottom.
Tilting down.
I wonder, is
Can
that
we look
some
down?
Hawthorne that's covered itself with rocks?
I don't think so, but I don't know. Could be.
I've never seen a little pile of pebbles like that down here anywhere, Shirley. And they're
Well,
little white
yeah.
pebbles. I mean, I think those
No
are
idea.
4M tests, the white things stuck
Yeah.
to whatever
Yeah.
that is.
I was
I just
looking at
wonder
that
if
too.
it's biology. And it's got a trail
Yeah.
to the left.
Yeah, so there's the
Okay, guys,
Yeah.
on the chat. Is
but
this a-
down below that, which was actually quite interesting, and a feature that Patty right there.
Right there?
That Patty pointed out earlier. Go over to the right
Copy that.
and zoom out a little bit more. Okay, now, okay. It looked like there's like a star-shaped pattern that was actually quite large, and Patty pointed that out earlier. Your lasers
I'm
are
drifting.
coming up on it, so it's just to the right of center.
There we go.
That's to the right of center.
And I'll tilt up just
And
slightly.
what she was
Well, we have
thinking.
Les Watling says, looks like something dumped their gut all at once. Debbie
Oh, well, that was
Blixen-
on the other one. I think that's what they're talking about, that
The
long thing.
pebbles.
Okay, so let's look at this. And if you'd
Okay.
like, just go zoom out just a little bit on that. What Patty was saying, when she looked at this, it looked like it was like five arms, and that maybe because Chris Ma was telling us the other day about how some
starfish do bury
sea stars
themselves.
can bury themselves. Whether that's what's happening here, I do not know.
I want to look at the pile of pebbles.
Okay, we're done looking at this. Pilot, our geologist wants to look at the pile of pebbles.
Copy that, the
I
one
think
that
the
we were
pile
looking
of
at a second.
pebbles is, yeah.
Sure.
I think that's a critter. And we
gonna
got a
jump up
couple
there just a
people
little bit.
down here saying it could be a holothurian. Hey, what I thought. Thank you, Scott. Oh, you made my day. That isn't unknown, he said, and it's got
Copy that.
Looks like
little.
it's
We're just
about
trying to replace
10
myself.
centimeters. It looks
Yeah,
a
it's
little
10
bit,
centimeters
about 10
long.
centimeters.
But it does have a trail.
It does have
behind
to
it.
the left, over to the left.
left.
It does.
Oh, but the star is real obvious
Yeah,
now.
I know,
It's
I know.
5 pennant. Got 5
Oh,
arms.
might come down too
oh, this
hard
is
there.
really cool. Mysteries of the deep.
Uh-oh.
Mike Ford says
a
maybe
filter for
nudge
that.
it. Everybody wants us to poke things. Leave them alone.
Go away, dirt. Let's get a good shot here.
Can we see anything
Yeah,
like
if we can
tentacles?
if we can zoom in on that, the
Real
right side,
close.
real close, we might be able to see some of
Oh,
the
yeah.
feeding tentacles.
Oh, golly, I can't
if,
see
If
anything.
in fact, if it's a holothurian.
But that's not a normal cluster of pebbles for down here.
It's really odd.
I'm sure that's a
something covered.
holothurian. Thank you, Scott.
And there's something on the surface of it as well.
Copy that. You can
I'm giggling reading the
take in some tether.
Check around.
the chat room. Sorry.
I don't think we put in a real large move, right? It was 30 meters? Okay. I can move forward a little bit after we're done here. Watch leader, are you okay if I go forward a little bit?
We still did not see
You wanna
that
go-
asteroid
sorry.
to the right.
Say again?
Are you okay if I go forward a little bit?
Yeah.
Yes, please.
Copy that.
Can
Watch
we-
lead nav.
Yes, Nav.
I just looked at all the numbers.
Mm-hmm.
We have to leave the
Flash
bottom
my
at
camera.
2:45 local, so we have 2 hours and 45 minutes left on the bottom. And we have about 580 meters to the final waypoint. So we'd have to average a little over 0.1 knots,
We
which
wanna
is
get
very
this white
doable.
object.
Right over here?
Nope, bottom right.
Thank you, Nav.
Down to the bottom right.
Skid.
If we do go slowly at the beginning, we can always, since it's so
Now
flat, we
straight
could
open
always,
lasers.
speed up at
Down
the end.
some?
Yes.
Once
Right.
there's
Right
your
there?
new species,
Oh, that little one?
Easily make the
Left of
waypoint.
lasers.
Got it.
Thank you very much.
You're welcome.
Okay, so we're doing
Not the white
great.
thing, but below that.
Yeah, right below me right now.
Yeah, it's right below the lasers. That's what we were looking at earlier.
And that's another thing you want to move in close.
Now it's just above
Tilt
the
your camera
lasers.
Down a little more.
Down?
Yeah, move in closer.
Oh,
with your
okay.
Camera
Yes.
tilted down.
Oh, so what are we looking for?
That-
I don't see anything.
No, you saw
Oh,
it
I
first
saw
before.
legs.
You saw it before.
I saw little white legs up there. I really do like the critters. I would love to see a bubbling spring at the top of
Yeah,
this
tilt
mud
the camera
volcano,
down
but
more.
there sure
I'm just gonna go
are
ahead and
a
get
lot of sediments here.
Landed over there.
Do I see
And we're gonna
legs?
land right here.
What is this?
Softly.
Very softly. Come on, softer than that.
Softly, softly, and not to kick up a lot of sediment.
Very nice.
Softly.
Ooh,
I see
well
it now.
done. Beautifully done, pilot.
Okay, here we go.
Now a zoom.
Go ahead, zoom.
Camera tilt down.
Tilting down. There you go.
Wow. Shirley's favorite
Okay, all
thing
right.
in
Now,
the world.
Boy, I'll
with
tell you
sediment.
what. If I had to guess,
This is a sponge.
my guess would be that it's a dead lithisted sponge. But
Greetings, co-pilot.
boy, I don't know. Chris, what do you think? Oh, Scott's saying. Oh, wait, hold on a second. Hold on a second.
You're
Scott
not roping
says-
me into this one.
Oh, no.
Okay, wait, hold on a second.
Okay, Chris,
Oh, wait,
read.
Allison's talking about.
Yeah.
Oh, wait, Allison
okay.
was talking about the holothurian. Okay, so Scott's saying foram
Agglutinating
and
foraminiferin.
agglutinating foraminiferin.
And Les
wow.
says, oh yeah, I think that's amazing.
Yeah, because it really wouldn't be a lithisted sponge like this.
Looks kind of like it though.
Well,
huh?
I know it does,
Sorta,
but it
kinda?
wouldn't. But yeah, but they're going to attach onto something.
Okay,
something hard
rather than just
yeah.
groove in the sediment.
Xeno, okay, sure, or Xeno504, close enough. Anybody want to tell us something more about this?
Yeah. Why does it agglutinate? In other words, stick all of that sediment to its surface. For protection?
These are single-celled amoebas like Protus. Xeno has forams on it. Wow. So actually, two
This
things
is amazing.
here.
I'm amazed that somebody spotted this thing against the background of the rest of the sediment.
Actually, you were the one who spotted it.
Did I?
Yes, you spotted it earlier, and you thought it was a
Got you loud and
squat
clear.
lobster, and then we got distracted.
Oh, right.
By looking at the
I saw the two
star-shaped
bottom legs.
the star-shaped pattern,
Yeah.
and then
Also,
once we
it's
were looking at the
flat as a
star-shaped
pancake
pattern,
out
we
here.
got
That's what we
distracted
zoomed
looking
on.
at the sea cucumber with pebbles on it.
I love the fact that there's so much stuff down
Right,
here.
and Allison Miller
And a
thought that it
starfish
might be that sea cucumber might be. Where am I? Going up the little chat log here. A Pseudostichopus or a
Straight
Molpad
to our starboard.
known to cover themselves in debris. That was about
Ooh, we got
the
another sponge.
sea cucumber.
Look at that sponge.
See it now, right
I
at
got
lasers on the edge of the
Shirley
map.
and Patty.
Yes.
Yeah, Scott, go ahead.
I was trying to get in, but I didn't want to interrupt you. That's why
I think the
said
ship
that
move is done,
sunglasses,
Sean.
symbol, so that didn't
Right.
work. But a Xenophyophore
Kinda looks like
is
it.
a single-celled protistan. It's comprised of one cell,
Yeah.
like the foraminiferans, and the way to sort of think about it is like an amoeba.
Okay.
And
So what do you want to look at, watch lead?
sediment grains, meaning it can glue together these sediment grains, and they take on
Not even
all
this
kinds
sponge?
of shapes and sizes.
I bet Chris Kelly wants to look at that.
We've seen many on
And
some
Shirley.
of the previous dives in golf ball shapes, but I think the reason
we'll
they're
take-
doing it is to make them
we'll take a quick zoom on the starfish for a little while.
organic particulates that are floating through the water column, and they use these little structures called pseudopodia, arm-like extensions of the cell, that they can bind up those particles, phagocytize, or take them inside the cell and digest them. So I suspect it's large and that shape in order to capture food.
Okay. Let's see. We can
And now we're looking at it's a sea star. Yep.
do a
Yep.
snap zoom. Hold there. Zoom in.
Oh yeah.
Okay. Going a little tighter.
There you go. Now it is.
Thank you. Okay, my mic was not open. Must have accidentally bumped it. But the sea star, the center of the body is so swollen. What I heard yesterday from Chris Ma from the Smithsonian is, I think it was Chris.
I think it was Scott telling us
Oops,
about it. Have you
Scott?
eaten?
Oh,
Its belly
yeah,
was
it was
full?
Scott telling us that it was full of food, and I wonder if it climbed up on the rock to sit there and digest in peace. Scott, if
Okay, you can zoom out.
you'd like to tell us something about this, that'd be great.
This is a different sponge. I wonder if we could get a close-up of this. I'm pretty sure
Certainly
you know
can.
what
Can we zoom on
phylum-
that sponge?
excuse me,
Certainly
family,
can.
Possibly genus it is, but
I do on the stock. Oh. Just gonna sit down real soft, try not to kick up sediment.
Okay, we're going to get a look at the bell of this sponge, and I wonder if there is something
Can zoom in.
kind of in the middle of the stalk that is white or
Well, these
light-colored.
typically have
Have
well,
things
The lasers.
yeah, oh,
There's
this
got
one
it.
a
looks like
couple of little amphipods
cyan.
or something. Anyway, something white.
Partial.
2 of them,
Going tighter.
I think.
Going to the white thing or the sponge?
Nope, just one. What is that on there?
Snail.
Oh, it's a snail.
snail. Beautiful.
Cool.
Yeah, yeah. Snail looks like we've got a polychaete and a
Wow.
tube.
That's
Yeah, actually some polychaetes and tubes we can see at the top. If you move, yeah, actually there's several polychaete tubes on there.
wow.
Chris, what are you thinking about this sponge?
Oh, it looks like a
I
tulip.
think it's highly matted.
Yeah, I do
And,
too.
I wonder if they could go all the way to the bottom with it.
All the way to the bottom.
So the stock looked more solid than I would have expected, but there seems to be a lot of sediment that's trapped in there that might be a result of the fibrous. Yeah, that's at the base. So I'm pretty sure this is a highly matted.
So,
What's on the
Nav.
seafloor behind it?
Another one.
I'm sure Levi's walking you through right now. Can you hear me?
Yeah.
Okay. So when you sit down at Nav, first couple things you want to look at
So
are
the sponges in this group are
and
very
surface conditions.
closely related to
So
a family
if we were to
called
lose
Farinamatidae, and we
DP
saw
on the ship, you know,
some
what
sponges
direction we
like
start
that
moving.
on one of the other dives. But they
also,
tend to have a fibrous attachment, and
if
it's a
you can
little
look at the average
confusing
pace
further up. But then you can
that we
see
need
it
to
sort of
make in order
dividing into
to
fibers right
get to the
here.
last waypoint.
The other thing
That'd be
about
great.
highly matted that
Calculate
makes
those now.
them very similar to the Farinamatids is they have that unusual microsclere, which is a microscopic spicule or skeletal part of the sponge that's called an amphidisc. And I described it as being 2 umbrellas that are hooked together by their handles, so the umbrellas are in opposite directions. And when you see that particular type of spicule, when you do a microscopic prep, you can eliminate an awful lot of other sponges and narrow it down to the Farinamatidae or the Hyalomatidae.
Thank you, Chris. That was Chris Kelly from University of Hawaii.
All right, watch lead. Well, we just had a shift change. So now we have Levi and Fernando over on Nav. Not sure if we're streaming it or not, but and then I'm Dan on pilot, and to my right I've got Sean on co-pilot, and just stepping into the chair is Tara Smithy in video, and behind us is Annie White, clipping out all this cool stuff we're seeing today, and we're all with the Global Foundation for Ocean Exploration.
Thanks, Dan. I'm Shirley Pomponi. I'm from Harbor Branch Oceanographic Institute, Florida Atlantic University, and NOAA's Cooperative Institute for Ocean Exploration, Research, and Technology. I am the biology science lead aboard the Okeanos Explorer, and Patty Fryer from the University of Hawaii is our geology science lead. It is 10 minutes after noon here, and she just stepped out to grab a quick bite to eat since we're seeing so much biology and we're seeing little things just scampering over the surface of this sponge. We are on the Okeanos Explorer, which is America's ship for ocean exploration, and we are coming to you live as we explore unknown areas of the Marianas Trench National Marine Monument. We're on an area now that we call the unnamed we call that the unnamed Four-Arc Mud Volcano. So all name suggestions are welcome. This expedition is only possible through a lot of partnerships and collaborations. The mission itself is designed and supported by NOAA's Office of Ocean Exploration and Research. The ship is operated by NOAA's Office of Marine and Aviation Operations. And the expedition is part of a 3-year campaign. The acronym for it is Capstone Campaign to address the Pacific Monument science, technology, and ocean needs. So this campaign was designed to address the need of our nation's National Monuments resource managers to better understand these deepest parts of our nation's marine monuments. We have a number of other partners, including several within NOAA. NOAA's National Marine Fisheries Service, the Marine National Monuments Program,
30
the Deep Sea Coral Research and
Let's do
Technology
40.
Program, and the National Center for Environmental Information.
What have they been doing? They've been doing
And you're
0.2?
hearing from
Probably
time
doing
to
little
time
hops.
from
Yeah,
our
about
shore-based scientists who
Yeah,
are
it's
also
been
very
steady
important
over 0.2.
collaborators
240 at 0.2,
in this
little hop.
expedition.
Yes, video?
We are now